Hello, my name is Ashley Thompson, and I'm with the Molecular Monitoring for Health and Environment Lab at Grand Valley State University. Our lab currently monitors wastewater for SARS-CoV-2 counts. However, within this, we are also looking at the pepper-mild mottle virus as a tool in wastewater-based epidemiology. Going into this, we had a couple objectives in mind. We wanted to develop a simple and cost-effective method for monitoring PMOV that was suitable for use in wastewater-based epidemiology. Additionally, we wanted to utilize PMOV counts as a fecal content indicator to help us normalize our SARS-CoV-2 viral counts that we were seeing in our samples. Now, wastewater-based epidemiology is a great field because many human pathogens shed through fecal matter, making it so we are able to monitor pathogens that otherwise would be difficult to monitor within our communities. However, because fecal matter varies between wastewater samples, it reduces our predictive utility of this methodology. However, normalizing fecal content may help, and that is where the pepper-mild mottle virus comes into play. This non-enveloped, rod-shaped, single-stranded RNA virus is the most abundant RNA virus found in human feces. It is 100K to 10 billion gene copies per single gram of dry fecal matter, making it extremely stable and helps us because it co-purifies with SARS-CoV-2. Now, compared to some other labs, we are doing something slightly different, and that is because we have developed a novel primer probe sequence in-house to help us reduce the costs it takes to run PMOV. Our primer probe is from the PMOV replicase gene, which is approximately 71 base pairs. We confirmed our assay to find that the amplicon sequence aligns 100% with NCBI PMOV replicase gene, increasing our confidence of using this in place of another primer probe option. Throughout this process, we have learned many things, and one of them has been that fecal content from wastewater treatment plant samples is relatively constant over time. All of these samples are wastewater treatment plants, and you see very little variation over time within these samples, making it extremely predictable on what the PMOV fecal content should be at a wastewater treatment plant. However, when you compare a wastewater treatment plant like GR in blue with samples grabbed from manholes like site VE in the red, you see a huge difference. While with GR, you are seeing that stable PMOV count over time. With site VE, you are seeing variables all over the place on what that PMOV content is going to be within a sample, making grab sites less predictable on what their fecal content should be. Within a grab site, you can also see variations in the samples depending on the sampling method that is being used to collect the sample to begin with. Site CE is a university that was monitored over the summer to determine how collection depth varies our PMOV data. What we found is that the deeper you sample, the more the virus has settled out and the more inhibitors that are likely to be present. So when the flow rate is lower, you are more likely to see those inhibitors and settled out virus. However, just because you're seeing more of this does not mean it is correlating well with the actual occupancy of a site with it. So in this graph, for example, we see that there is a spike in the number of occupants on this campus around June 5th. Yet on our lowest site, sampling site, CE3, we see a drop in PMOV due to that higher flow rate of the occupants. So when you go to collect a sampling site, you have to consider what depth you want to collect at. That's going to give you the most accurate idea of what your current population is experiencing. Throughout this, we have learned so many new things, and we are extremely grateful for that. What we've done is successfully design our own primer probe combination to effectively quantify PMOV in wastewater samples in either the qPCR or the ddPCR platform. Fecal content in wastewater treatment plants is found to be relatively constant over time versus our grab samples from manholes, which are more likely to be variable. Monitoring PMOV enables a greater understanding of the dynamics of the virus movement and the inhibitors in sewage that are contributing to more consistent grab sampling procedures. We have used PMOV monitoring in our own lab to identify outliers to ensure the quality of both our sampling and processing consistency. And when it comes to our manholes, we have been using PMOV to evaluate our sampling locations with respect to their fecal content to one, help us identify outliers, and also to determine what range of variability we should be seeing at a site. And when it comes to all of our samples, PMOV has been a great way to identify any sources of contamination, especially in your negative control or extraction controls, where it might be missed in other platforms. Based on all of this and our experiences, we have determined that PMOV may be a good candidate to normalize SARS-CoV-2 counts in wastewater-based epidemiology. I would like to give a huge thanks to Grand Valley State University's Cell and Molecular Biology Department, the Molecular Monitoring for Environment and Health Lab, and our team members, MDHHS, Kent County Health Department, Grand Rapids Water Resource Recovery Facility, Wyoming Clean Water Plant, Granville Clean Water Plant, Cedar Springs Wastewater Treatment Plant, Brightway Plumbing, and Preen and Newhoff. Thank you very much.